Now we understand all this, we can understand all the main types of non-covalent interactions between molecules in biochemistry. Dipole interactions and hydrogen bonds, charge-charge interactions or ion pairs, van der Waals forces, and hydrophobic interactions. From the previous examples, you have seen that bonds of carbon and hydrogen with the highly electronegative oxygen and nitrogen are always polar. Picture these situations. A hydrogen bound to either oxygen or nitrogen is face-on to another oxygen or nitrogen. The partial charges attract each other, and you have a hydrogen sandwiched between the two more electronegative atoms. The non-covalent bond between these two atoms is called a hydrogen bond. The atom that is covalently linked to hydrogen is called the hydrogen donor, and the one that is non-covalently linked, hydrogen acceptor. A hydrogen bond can only form if the hydrogen and the acceptor are about 0.18 nanometers apart. That's about double the length of the covalent bond. It is also important that the three atoms involved are all in a straight line. There is some room for deviation here, but the strength of that bond decreases quickly the further away from the ideal constellation we are. Probably the single most important hydrogen bond in biochemistry is that between water molecules. You can see that the two molecules need to be arranged in a particular way. In this 3D version, the two pairs of black dots here show the electron pairs of the oxygen atom that are partially shared with the hydrogen atom to make this non-covalent hydrogen bond. Because there are two hydrogens per molecule of water and two pairs of unbonded electrons on the oxygen, each water molecule can form hydrogen bonds to 4 other water molecules, 2 as hydrogen donor and 2 as acceptor. All 4 of these possible bonds are only realized in ice, which results in a hexagonal lattice. In liquid water, the molecules are too restless to align in just the right way with 4 other molecules, and instead they jiggle about. Hydrogen bonds are formed and broken all the time, and they're flickering on and off, and on average don't last longer than 10 picoseconds. When water freezes, these hydrogen bonds make the water molecules space themselves a little further apart than before, which is why ice has a lower density than water and floats. Still, there are lots of these hydrogen bonds in liquid water, and breaking one takes about 20 kilojoules per mole. Among other things, this explains why water has a very high boiling point, and it also explains hydrophobic interactions, a different kind of non-covalent interactions, as we're going to see later. In biomolecules, there are really only 2 main types of hydrogen donors, the amino group and the hydroxyl group, and 3 different types of hydrogen acceptors. All of these are common in biomolecules, and hydrogen bonding is essential for the 3D structure and stability of DNA, proteins, and some polysaccharides. Have a look if you can find the hydrogen bonds that hold the guanine-cytosine pair and DNA together. Guanine and cytosine form 3 bonds, as you can see. Adenine and thymine form 2. Peptides and proteins also form hydrogen bonds that are essential for protein structure. Try and find the hydrogen bonds formed between these 2 peptide strands. We'll get to these bonds in greater detail in a later lecture. So let's summarize. Hydrogen bonds are one of the most common types of dipole-dipole interactions in biochemistry. Of all the different types of non-covalent bonds, it is one of the strongest ones, and it takes about 30 kilojoules per mole to break one. That's still more than 10 times weaker than the strength of a covalent bond. Hydrogen bonds can form when a hydrogen atom bonded to an electronegative atom is attracted to another electronegative atom with lone electron pairs. X and Y here are practically always oxygen or nitrogen. And finally, Hydrogen bonds are only strong if all three atoms are close to a straight alignment.